morning, everyone. I'm Eric Nestler. I'm director of the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai in New York City, and I have the distinct honor of serving as co-chair of the Scientific Advisory Board for One Mind. Today, we have the honor of introducing one of our Rising Star Award winners, Dr. Ignacio Saez, who, by chance, is also from the Icahn School of Medicine at Mount Sinai, where he's a rising associate professor in the Departments of Neurosurgery, Neurology, and Neuroscience. Ignacio, welcome.
Thank you. I'm glad to be here.
And congratulations on the Rising Star Award.
Thank you so much.
It always gives me particular pleasure when one of our own wins this award.
It's a huge honor.
Yeah. We wanted to talk today about the research that you're proposing as part of the Rising Star Award application. And I gather from your work that you use the human brain to do your experiments. We come up with the term the Human Brain Laboratory, so individuals who are undergoing neurosurgery, and then you work with those individuals to examine how their brains are functioning. So that sounds like a lot.
Yes.
Tell us more about that, please.
Yeah, I'll start with a little anecdote. When I started my academic career in neuroscience, I actually work on animal models. And one of the main reasons why I did that is because we have such powerful techniques to measure, record, and stimulate the electrical activity of individual neurons or pieces of the brain. And I was really drawn to that. But with time, there was a growing discomfort in me about the multiple layers that we have to cross between those in vitro studies and the actual application of the acquired knowledge to treat patients. And that I knew that my heart was somewhere close to patients and clinical settings. And for the longest time, I didn't see a way in which I could apply my methods and my techniques to the study of the human brain, because these are just invasive methods that are not applicable to your everyday patient or person. But at some point in my career, the opportunity opened up to do exactly that, which was kind of a mind-blowing moment for me, to know that I could use those powerful techniques and use them to study the human brain, human thoughts, feelings, and actions in a principled and ethical manner. And the reason that is possible is because we do have surgical interventions in the clinic that are used for the diagnosis and treatment of a variety of disorders, and this is happening in hospitals all over the country, very often at Mount Sinai, where we have epilepsy patients and Parkinsons patients and depression patients that come in and undergo surgery for clinical purposes.
So let me just back up a moment, make sure our listeners are appreciating this, because I do. My laboratory studies rodents, mice and rats, and in my laboratory, like in many others, we have the ability to put an animal under anesthesia, drill a hole in its head, lower electrodes into the brain of that animal, and then have the animal wake up and run around a maze and perform various tasks. But we're always limited by the ability of what we can infer from what an animal is doing. You're telling me that now, because these individuals are undergoing neurosurgery, so they already have the opening in their skulls, electrodes in their brain for therapy, that you can actually do the same experiment in a person, only now the person can talk to you while you're doing your recordings. Did I get that right?
Basically, yeah, and it makes for such a massive difference in some respects, because we have access to that patient's train of thought and their internal feelings, and we can just ask them questions which are impossible to ask of animal models, right?
Yeah.
So that is.
Yeah. Can you give us some examples of that?
Yeah, for sure. So, so we work. One of the main projects that we have ongoing in the lab is the application of some of these techniques to the study and hopefully developing new treatments for depression, which is characterized by altered patterns of thoughts, feelings, and behavior. Behavior we can measure, right? Behavior can be measured in animals also. But the opportunity to ask a patient point blank, how are you feeling in this moment, right? And them answering, however complex and long the answer is, but they actually give you a snapshot of their internal state, which is otherwise impossible.
Happy, sad, anxious, anticipating. Yeah.
Yeah, exactly. And so it gives us access to that internal milieu and their feelings in a way that is super, super powerful.
And so tell us about some of the findings that you've had so far.
Right. So my lab works on both studying the neurobiological basis of human behavior, especially decision-making behavior, and also on the translational applications of those, because decision-making behavior is changed and damaged in a variety of disorders, including depression. So some of the things that we've observed is that even though sometimes we think about specific brain areas carrying out specific calculations that translate into behavior, we've been able to show that there's a real network type activity, which is not just a single region, but multiple regions at the same time that act in a concerted and coordinated way. They're not doing identical computations, but they are doing related things. And so they coordinate at a very fast pace, very quickly, because if you think about it, when you make a decision, that process can be very fast. It could be sub-second, right? You see your options, and then you make a snap decision, right? So that's one of the powers of the technique.
So you're recording electrical activity.
That is correct.
From individual cells or clusters of cells?
Clusters of cells.
Clusters of cells.
And individual cells.
And individual cells. And you're trying to then correlate activity patterns of those cells with a behavior, with a feeling.
Right. Yeah, maybe I should explain that a little better. But we are implanting, or the surgeons are implanting, tens, dozens to hundreds of electrodes in the brain of these patients. Every one of those electrodes is a small circular metal piece that basically reports as a voltage the coordinated activity of hundreds of thousands to millions of neurons in the vicinity of the electrode tip.
And this is helping the surgeon to know where to do his surgery, right? So this is part of the treatment for the patient.
Exactly. Yeah. In the case of epilepsy, this allows the surgeons and the neurologists to examine that brain electrical activity and find where the aberrant activity that gives rise to seizures is coming from in a highly precise and anatomically correct manner. And so that's the reason why the electrodes are implanted, and we're just leveraging that opportunity to take those recordings and ask questions. These patients are actually under observation in the hospital for days or weeks. And they're just waiting to have a seizure or two or three, because that is the reason they're in there, right?
And they need to be in the hospital because a portion of their skull has been removed, as I understand it?
So sometimes it's not a whole portion, but small drill holes, but they do have wires coming out of their head, and they're connected.
For safety, it's necessary to stay in the hospital.
Yeah, so there are limitations, right? We were talking about the animals running around and so on. Some of those things we can do. The patients are bedridden and then they can't move, but they're humans. They're really smart, so we can have them play a computer game instead, right? Where they navigate a virtual environment or do whatever it is. And that reminds me of something else that I think is important, which is when we go in and we instruct these patients to play a particular game or carry out a specific task, the instruction is so fast. We go in there and we verbalize it, and within 2 minutes they're doing it. So they're really tapping on their natural networks and their natural behavior to carry out these tasks in lightning fast, kind of one-shot learning.
And I understand for the epilepsy patients, at least, that they enjoy working with you, because otherwise they're just sitting in the hospital bored stiff most of the time, right?
Yeah, definitely. I mean, we do try to engineer our tasks in a way that's fun. Right? We try to gamify them so it captures their imagination. They want to do it. But most patients are both extremely generous with their time. They're very conscious that we're trying to do something for them in the long run. I've had patients tell me these. When we explain clearly this is not going to benefit you or your clinical, and it's not going to inform your clinical treatment, I got this response from patient one that said, I don't mind, I'm here because someone volunteered 20 years ago, which was a very touching response. But yeah, they do enjoy it. It helps them pass the time. They engage with my research team often, and they like playing these games. We banter a little bit, and they're generally great. And it really helps us understand the ways in which they work.
So then, going back to the therapeutics of things, the recordings that you obtain, and then you would use various artificial intelligence, machine learning algorithms, I presume, to associate electrical activity of the brain with behaviors. You would then use that to inform mechanisms of what's going wrong, say, in a person who has depression. So tell us about the therapeutic context of where you do your work, deep brain stimulation used for a treatment for depression. Tell us a bit about that, please.
Yeah, exactly. So the opportunity is twofold. One is this recording that we've been talking about, where we're just passively measuring electrical activity in all these brain regions that have received an electrode, and that is very valuable. But at the same time, there's an opportunity to modulate those neurons that are in the vicinity of the electrode by doing a very carefully controlled and small, passing a little bit of current in those electrodes. That activates the cells in the vicinity, and it actually changes the way that the brain
That would
works.
be something akin to a cardiac pacemaker, only instead of the electrode going to the heart, the electrode is now going into this place in the brain, right?
Exactly.
Yeah.
And that is absolutely the way that it's a great way to think about it, because in the case of deep brain stimulation, which is routinely used in many hospitals all over the world, there's over 200,000 patients implanted with these DBS electrodes in a safe and effective manner. These patients go back home and they have the electrode chronically implanted. They live with it. They, once you go through the surgery and the hospital stay, it's easy to forget. You're carrying those electrodes, and it really helps them manage their symptoms. At Sinai, we also have clinical trials where we're trying to develop new interventions for these operations, where we're trying to increase the range of pathologies that can be treated effectively this way. And this is not just work from my lab, but work from other labs in Sinai, like Dr. Mayberg and others. But the idea is that we have these therapeutic intervention, this deep brain stimulation, this brain pacemaker that is extremely effective with a handful of diseases, including Parkinson. And there's a mixed feeling of frustration and hope that we can use it to treat other diseases, right? If it's effective for Parkinson, why shouldn't it be effective for OC, obsessive compulsive disorder, depression, anxiety, etc.?
And the FDA has given its approval for Tourette syndrome and obsessive compulsive disorder, and you and your colleagues are looking to extend that to depression and many other syndromes.
Exactly. And I think what the way I think about this is that the reason we don't have those other applications is because we just don't understand enough about how the brain of a depressed patient is different from the brain of a non-depressed or healthy individual, right? And so we're trying to backfill that knowledge in a very targeted way to understand how is this brain different from this other brain, and can we use those powerful stimulation abilities to recover the activity and bring it back to a healthy state.
Yeah, no, it sounds amazing. And as I think about the field of neuroscience, I think the ability to use the neurosurgical suite where an individual is having surgery for treatment to learn these deep fundamental features of how the brain is functioning is incredible. And I think really promises tremendous advances for the future. AI and machine learning are everywhere in the news today. And I take it that your laboratory makes heavy use of those approaches, because as you obtain information about the electrical activity of nerve cells in the brain, that's a lot of data. So just explain in a way that I'm not a mathematician, explain to me in a way that I could understand how AI and machine learning is helping your research.
Yeah, so it is absolutely correct that we now have a good problem that we used not to have, which is massive amounts of data coming in through our electrodes and our recordings. When I was a PhD student, I would record from one cell at a time, maybe two, and that was already exciting and novel. But now we're recording from millions, right? Hundreds of electrodes, incorporating the activity of millions and millions of neurons at the same time, right? So that creates...
And a human brain may have 100 billion nerve cells, but to record from 1 million cells at a time is incredible.
It is incredible, and it is extremely powerful, but it creates this problem that, how do we analyze, interpret, and understand this massive amount of data that's coming in? It's not trivial. If you only have, the one cell to look at, you're going to examine it from all possible angles, and eventually you'll figure it out. But when you have millions and millions, how do we deal with that? And machine learning approaches and AI approaches are one way in which we are effectively querying this data to make sense of it. And so just to give you an idea of the kinds of things that we're doing in the lab, we like to talk about brain states. So not just the electrical activity in brain area A or brain area B, but the aggregate activity over these many areas that reflects something that is either happening in the external world or that reflects a way in which the patient is feeling or thinking, and so on and so forth, right? So, for example, we touched on our work on decision making. We have now implemented machine learning and AI models that can take that electrical activity from those hundreds of electrodes simultaneously and can predict with pretty high accuracy on a trial-by-trial basis what the patient is going to choose to do a few hundreds of milliseconds later. So before there's an overt expression of the choice, we know what's happening because we're getting all that data, we're running it through these models, and it's eventually giving us the answer.
Right. And I know that that's one of the biggest questions of our time, and that your work is contributing importantly into decoding how it is that these millions of brain cells are computing problems and making decisions.
Yeah, and it's a powerful approach, among other things, because it's flexible, right? We started using it in the context of these decisions, but a brain state could be defined in many different ways, right? It could be, a thought, a feeling, a memory, or it could be a pathological state. We talked about neuromodulation. A brain state could be a depressed brain state versus a healthy brain state. And we're tapping onto those methods to help us understand what those states look like and how to change one into the other.
Right. We know that conquering brain illnesses will be the last frontier of modern medicine. The brain is just so much more complicated than every other organ in the body. Yet it's so critical for people with brain disorders or their families to have hope. And I remain as optimistic as ever that we're going to conquer these illnesses. It will just take the kind of fundamental research like you and your colleagues are doing. So thank you so much. It's great to have you join us for this scientific symposium over this weekend, and best of luck with your important research.
Thank you very much.